Great.
Can you send
Thank
it?
you very much, and good morning, everyone. My name is Augustine Obour with Kansas State University. I'm based here at the Agricultural Research Center in Hayes. Today, I'll be sharing with you a little bit about ion chlorosis mechanisms in high pH soils. As an outline for this presentation, I'll talk briefly about reactions of ion in the soil, some mechanisms of ion acquisition by plants, and then causes of ion deficiency chlorosis in the soil, and then how you can manage a little bit about management of ion deficiency chlorosis in high pH soils. So as we all know, iron is a very important nutrient for plants. In the soil, we have a lot of iron in the soil. However, the forms that is present in the soil, the solubility is very low. So that is why we tend to have iron deficiency in high pH soils. Basically, we have four main forms of iron in the soil. The iron in the soil can be in the reduced form, that is the ferrous state, which is present in primary minerals and some phyllosilicates. We can also have the oxidized forms of iron, but in the soil it's mostly present in the hydroxyl or oxidized forms of iron. Then we also have exchangeable forms of iron, and then these are in the soil solution. Then we also have iron bound to organic matter. So in calcareous soils, however, all these species of iron, the solubility is very low. It's around 10 to the power minus 10 moles. This is relatively very, very low compared to how much it should be in the soil for plant uptake. So if the plant doesn't have any mechanisms to solubilize this iron, then when you grow them in high pH soils, the possibility of ion chlorosis is very high. So I'll talk a little bit about functions of iron in the plant. Iron is very important in chlorophyll synthesis. So that is why we tend to see yellowing of plants when we have iron deficiency in the soil. Iron is also very important in protein metabolism. So if you have iron deficiency, we tend to have decreased protein contents in plants. Iron is also very important in many proteins and enzymes that we find in the plant. For instance, we have cytochrome oxidase, which have some components of it, the constituents are coming from iron. We have peroxidase that helps in reduction of iron, 3 chelase, which is also, iron has play a very important role in this. Then we have ferredoxins that are important in the electron transport systems and nitrogen fixations. So we see the importance of iron to the plant is because it's involved in so many enzymes and proteins formations in the plant. Iron deficiency, like I said, is a problem in high pH soils because the iron is very low in solubility because of the forms that we have, like the oxides and hydroxide that are low in solubility in high pH conditions. We tend to have iron deficiency in these soils. This is a picture that I took in Western Kansas sometime in 2018. Sorghum showing iron deficiency chlorosis because of limited iron availability for sorghum uptake. And most of the time, iron deficiency normally occurs in the younger leaves because iron is relatively immobile, so it doesn't move from older leaves to younger leaves. So we tend to see interveinal chlorosis of the younger leaf. This is a common visual symptom of iron deficiency chlorosis in high pH soils. So how does iron react in the soil, iron availability in the soil? So the availability or solubility of iron is controlled by many chemical and biological processes in the soil. The best equation that sums up all these reactions in the soil is this one here. So we have iron in the soil, mostly in the form of oxides, and these oxides can be reduced to the cationic form, that is iron 3 in the soil. And this is controlled by reactions in the soil like pH and oxidation and redox processes in the soil. For instance, if we take soil pH, for example, under high pH conditions, the soil tends to be very alkaline. So under alkaline conditions, there's a lot of hydroxyl ions that are produced in the soil. When you have hydroxyl ions, it combines with the cations and then it forms iron hydroxides, or it forms iron oxides. These iron oxides are very insoluble compared to the cation form. So that is why in high pH soils, we have very little iron available for plant uptake, because the forms of iron changes from the cations to the oxidized forms of iron. However, when we go to lower pH conditions, is the reverse situation. We have the oxides which are insoluble, they are converted to iron 2. So under these conditions, we have a lot of iron present. The solubility of iron increases. Iron 2 is more soluble than iron 3, or the oxide. So under low pH conditions, we tend to have more iron in the soil because of this process. This is a figure that is showing the pH effect on micronutrient availability. On the x-axis is soil pH. On the y-axis is micronutrient uptake. In this case, zinc and manganese uptake in wheat. As you can see from this figure, when pH is around 4.5, we tend to have a lot of the micronutrients present, zinc and manganese in this case. We don't have iron in this figure, but if iron was to be here, it will be kind of the same characteristics as zinc. So as the pH increases, we tend to see a sharp decline in zinc or manganese or iron concentration. So as we have a decrease in the concentration, the amount of iron or zinc or manganese that is going to be taken by the plant is also going to be reduced. As I mentioned previously, as the pH changes from acidic to alkaline conditions, the ionic forms of the micronutrients, in this case iron or zinc, also changes to the hydroxyl forms or the oxide forms, which are less soluble. And this results in decreasing the solubility of the micronutrient in question, and that affect availability for plant uptake. The other reactions in the soil that affect availability of iron is oxidation reduction processes in the soil, which is also mediated by microorganisms present in the soil. So first one we talk about is under reduced conditions. For instance, when the soil is flooded. So under reduced conditions, iron 3 or iron oxides are reduced to iron 2. So when you have that kind of condition, then availability of iron increases because of the reduced state. So it reduces from iron 3 or iron oxides to iron 2, and that increases the availability of iron for plant uptake. However, under well-drained conditions, we tend to have the iron oxides being reduced to iron 3. Being reduced to iron 3, you get the oxidized form of iron, and this oxidized form of iron is highly insoluble compared to the reduced form of iron, and that reduces iron availability for plants. This process is, like I mentioned, is mediated by microorganisms in the soil that can use iron in the soil, and then it can either cause oxidation of the iron or reduction of the iron. So these processes have significant impact on solubility of iron in the soil. What is the mechanism of plant acquisition of iron? Different plant species differ in their ability to absorb iron from the soil. Basically, we have two main strategies that plant uses in acquiring iron from the soil. The first one is strategy 1 plants, which will be our dicotyledonous plants or non-gramineous monocots. And then we also have our strategy 2 plants, which will be our grasses. So in strategy 1 plants, the main mechanisms involved is the ability to reduce the iron oxides to iron 2 to increase the availability for plant uptake. So what these plants does is they can release hydrogen ions into the soil rhizosphere around the roots. So by so doing, the soil pH is reduced. And as pH decreases, it favors the formation of iron 2, which increases the solubility and availability of iron for the plant uptake. They can also release chemicals, chelating agents that are capable of forming a chelate with the iron 3 or the oxides in the soil solution. And this is able to be carried across to the membrane, and then it reduces into ion 2 for plant uptake. They can also increase production of organic acids, example citrus. Once organic acids are produced in the rhizosphere, this decreases the pH around the roots, and that favors the conversion of ion to ion 2 to help in uptake by plants. Examples of plants in this that uses strategy 1 include peanuts, soybeans, peas, and then tomatoes. We also have strategy 2 plants. These strategy 2 plants are mostly our grasses like rice, wheat, corn, and sorghum. Here, the mechanism is being able to solubilize iron by using phytosiderophores. So these phytosiderophores are organic complexes that forms complex with iron, ion 3 or the iron oxide in the soil. This phytosiderophore ion complex then moves across the root membrane into the root cells. Once they are in the root cells, then the ion can be reduced into ion 2 for plant uptake. This is the strategy that the grasses uses. However, to be effective, you have to produce more of these phytosiderophores, so you will be able to have more ion for that plant for uptake. So there are differences in this strategy 1 plants and strategy 2 plants, how they move these chelated complexes or making ion available for them. So this is what is demonstrated in this figure. So the figure to my left, that is figure A, will be a strategy 1 plant like soybean. So let's look at this. In terms of ion present in the soil, it may be in the form of ion 3, in the cation form, or it can be in the oxide forms. This ion is then complex with the organic molecules to form a chelate. Once this chelate moves to the root membrane, at the membrane, this strategy 1 plants are able to reduce the ion chelate, and then the chelate move into the soil solution. And then the reduced ion then moves across the membrane into the root cells. Once the chelate material moves into the soil, it can bind with another ion molecule, and the process is repeated. However, when you come to the figure on my right, which is the strategy 2 plants, like the monocots like sorghum, the same process, you have ion 3 in soil solution, you form combines with a chelating agent, form a chelated ion 3. However, the difference is when it gets to the membrane, instead of being reduced, the whole chelate and ion complex move across into the root cells. Inside the root cells is where the ion is reduced to ion 2. And then the chelated material, the chelate, then move all the way from the root cell across the root membrane back into the soil solution. Once the chelate is in the soil solution, it can bind with another ion, and the process is repeated. Look at the difference here. The distance that you have to travel, you go all the way to the root cells and then come back into the soil solution. So what it means in terms of management is that the plants that uses strategy 2, you will need more of this chelated product or chelated agents to be effective compared to a strategy 1 plant that the reduction occurs at the membrane. So the chelate can get back in solution, and then the process is repeated again. So I would like to talk a little bit about causes of iron deficiency chlorosis in plants. There are soil factors, there are nutrient factors, and there are also environmental factors. The first soil factor is soil pH. We have talked about this previously. The solubility of iron decreases significantly when pH increases. pH above 7, solubility of iron decreases. For every unit increase in pH, for example, iron 3 activity or concentration decreases by 1,000-fold. And then for ion 2, the activity decreased by 100-fold with an increase in a 1 unit increase in pH. So in terms of management, if you are able to reduce the pH even by a half unit, that is very significant, because that is going to increase the solubility of iron and then make it available for plant uptake. Then there's also calcium carbonate, lime-induced chlorosis. When you have a lot of calcium carbonate in the soil, this calcium carbonate under hydrolysis releases bicarbonate ions in soil solution. Once you have bicarbonate ions, it creates alkaline conditions that favors the conversion of iron to iron 3 or to iron oxides. These iron oxides are very insoluble, so iron availability to the plant is decreased. Then we also have soil test levels, soils that you can have very low iron concentrations in the soil. So in that case, the availability for plant uptake is very low, and there's the tendency to cause iron deficiency chlorosis when you grow susceptible plants in this kind of soil. Then we also have nutrient interactions in the soil. The first culprit is phosphorus. High soil phosphorus in the soil can decrease iron availability because iron reacts with phosphorus to form iron phosphates. So in soils, high pH soils, if you have a lot of phosphorus in the soil, it can actually reduce iron availability. Then we also have nitrogen. Nitrates, plants take up nitrates and then they release, in exchange, they release bicarbonate ions into the root zone. If that happens, we know that bicarbonate causes alkaline conditions in the soil that decreases the solubility of iron. So if you have high nitrates in the soil, there's higher possibility of decreased iron availability for plant uptake. On the other hand, if you apply ammonium-based fertilizers in these high pH soils, this ammonium fertilizer has to be converted to nitrates, and in the process, hydrogen ions are released. These hydrogen ions can decrease the soil pH just around the root zone, and this can help to increase iron availability for the plant. Then there's also antagonistic effects between interactions with zinc and manganese. So if you have high levels of zinc or manganese in the soil, that can also affect iron availability for plant uptake because there's going to be some competition between these micronutrients. Then there are environmental factors like soil temperature. So if you have very low temperature, it can reduce plant growth. Root growth is impacted, and that can affect the ability of the plant to take up iron from the soil. So it can lead to iron deficiency. Then there's the possibility of under low temperature conditions, you have increase in CO2 solubility, and that can increase bicarbonate formation. If you have a lot of bicarbonates resulting from CO2 solubility, that can also affect iron availability for plant uptake. Then low temperatures can also affect phytosiderophores formation. So if you have phytosiderophore formation impacted, then that can affect iron uptake by strategy 2 plants. Then we also have high temperature conditions that in the soil can also affect microbial activity. You have increased microbial activity, they will produce more CO2, and that can result in bicarbonate formation, and that can affect availability of iron. Then we have soil moisture. If you have high soil moisture in the poorly drained calcareous soils, obviously you are going to have high bicarbonates in the soil that can impact availability of iron. On the other hand, in reduced conditions, we can actually increase iron availability too, because under reduced conditions, the form of iron that is favored is iron 2, and this is soluble than iron 3. Actually, under flooded conditions in some areas, we can have iron toxicity occurring because of this process. These are some pictures from our work that we did in Western Kansas in grain sorghum. We have water helping to improve iron availability for plant uptake. We have some research under dryland in Garden City and then under irrigation in Tribune. These pictures are the same hybrids that we planted under dryland. Seeing that on my left side, we have severe chlorosis of grain sorghum, the same hybrid. But under irrigation in Tribune, even though we're seeing some deficiency, it's still doing better than the dryland condition. And there are two things happening here. Just the water can help improve growth, the small roots that helps improve ion uptake. But also under irrigation, the form of ion is going to be ion 2 compared to the dry land where we have ion 3. So this the ion 2 is more soluble, so we have more ion available for plant uptake in the irrigated system than in the dryland environment. So how do we manage ion deficiency chlorosis in high pH soil? I'll talk a little bit briefly on this one. We can apply amendments like iron sulfate, but this doesn't work very well because iron sulfate reacts with bicarbonates and becomes unavailable. So you need to apply large amounts of these to be effective. What is more effective is the application of ion chelates. Ion chelates are more effective because the ion is protected from being from reactions with other molecules or carbonates or bicarbonate, other things in the soil. So we have different forms of ion chelate. Not all of them are effective on high pH soil, especially the EDTA forms are not very stable compared to ion EDDH forms of the chelate product. So this figure is showing just the molecular structure of ion EDTA and then ion gluconate. These are all chelates formed in both structures. The ion is being protected. See, we have the ion here is being protected in the structure, but it becomes available for the plant. So these chelates are able to increase the availability of ion or other micronutrients for plant uptake. So I mentioned that under high pH conditions, not all the chelates are stable. Some of them, their stability decreases under high pH conditions. And this is what this graph is talking about. Here on the x-axis is the number of days following the chelate application. And then the y-axis is the amount of the chelate in solution. You can see all these EDTA forms from day 0 after application. We have significant amount of it in solution. However, after 10 days of application, you see the EDTA forms, the stability is very, very low. It's breaking down. So the concentrations of these micronutrients in solution in the chelated form is very low. However, if you look at ion EDDHA, on day 0 is very high. Even by day 20 or 25, there's still a lot of it in solution, showing that these forms of ion chelate are fairly stable under high pH conditions. We conducted some experiments looking at the ability of using ion chelates to control or manage ion deficiency in grain sorghum. So these are some pictures from our work. Here is a sorghum hybrid 85Y40. This is a pioneer hybrid that is showing significant chlorosis. There's significant ion deficiency in this variety, this hybrid of sorghum. However, when we apply ion chelate, here we apply 3 pounds of the product per acre. We saw a significant improvement in the reduction of ion deficiency with the ion chelate applied. Then there's also differences in hybrids. Some hybrids are tolerant to ion deficiency compared to others. So these are 4 different hybrids. This is NK5418 that we did not apply any chelate. It's showing some chlorosis, but not as bad as Pioneer 85Y40. So we have 87P06, which is also showing some tolerance to ion deficiency chlorosis. Pioneer 86G32 also showing some tolerance. However, Pioneer 85Y40 showing significant chlorosis. So there is differences in hybrids. So some of the management that you can do is selecting hybrids that are tolerant to ion deficiency. This is grain yields from the same plots that we harvested in 2015. Here I'm showing the yields of the various hybrids on the x-axis are the hybrids, and then the y-axis is the grain yields. The green bar is the control where we didn't apply any chelate. The red bar is where we applied 3 pounds of the product. And then the green bar is 6 pounds of the product per acre. And then the purple bar is the 6 pound that was split, applied. As you can see, some of this first hybrid, NK5418, we did not see any difference between where we applied the chelate and then the control. However, if you look at 85Y40, Pioneer 85Y40, there's significant improvement with iron chelate application that helps to improve grain yields. Then this is grain yields from 2 of our dryland sites. On the x-axis is our iron sorghum hybrids, and then the y-axis is our grain yields. The blue bars are yields that we harvested from our research station, and the orange bars are yields from the producer sites. As you can see, there are differences between the hybrids. Some of the hybrids like 86G32, which shows some tolerance to iron chlorosis, have very high yields. However, we are also seeing significant differences between the 2 locations. They were 3 miles apart. What is happening here is the difference in the soil conditions. So this is the soil test results from the 2 sites. So our experiment station in Garden City and then the producer field. The soil pH is not different. Soil organic matter is not different. Iron concentration were similar between the 2 sites. However, if you look at the soil phosphorus levels, at our research station, we have about 26 ppm soil test phosphorus. The producer site has about 63 ppm phosphorus. And then when you look at the calcium carbonate percentage, we have 2.5% at our research station. And then the producer side has about 8% calcium carbonate percentage. So what happened is the high pH conditions with high soil phosphorus is going to reduce availability of phosphorus, and also, more importantly, the calcium carbonate levels. So this field showed severe chlorosis compared to our research station. The plots at the research station were actually able to grow out of the chlorosis, but not all the hybrids grew out of iron chlorosis at the producer field. Well, I think this is my last slide, so I'll end here. And thank you very much for listening. If you have any questions, you can contact me at this email address. Thank you.
Thank you, Dr. Obour. Does anybody have any questions for Augustine before we move on to our last video?